Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Challenges of Developing Antidrug Antibody and Concentration Assays for Lysosomal Transgene Enzymes on an Automated Immunoassay Platform. I'm Roisin McGuigan, an editor at BioInsights, and joining me today I have John Chappell and Issa Jamunbandi, who will discuss approaches for monitoring the concentration of expressed transgene products and any associated immunogenicity. So first, I'd like to introduce our presenters. John has around 25 years of experience in the contract research industry, supporting both preclinical and clinical drug development. He now leads the application support and service teams for Gyros Protein Technologies, where he is responsible for customer service and technical support in Europe and the Asia Pacific regions. Issa has over 10 years of experience developing and validating antidrug antibody, neutralizing antibody, and PK assays using various platforms, including the MSD, GyroLab, and various absorbance, fluorescence, and luminescence platforms. He is a subject matter expert in the development and validation of ADA and neutralizing antibody assays for challenging therapeutic products, ranging from small peptides and bispecifics to AAV gene therapy and transgene products. So after today's presentations, we'll have a live Q&A session. You can pose your questions to John and Issa at any time throughout the event using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. And if you'd like me to direct your question to a specific presenter, please do let me know when sending it in. I'd also like to draw your attention to the Resources tab, which is on your right, where you can find more information on the topics we'll cover today. And so without further ado, I'll hand over and we'll get the presentations underway.
Welcome everybody to this presentation. Today I want to give you an introduction to the GyroLab platform. My name is John Chappell and I'm Director of Scientific Support for both Europe and Asia, and I'm very much responsible for supporting customers, ensuring that their assays are working properly on our instrument. Today I will give you an introduction to Gyros Protein Technology, a little bit about the company and how we operate, and also an introduction to the technology, the basis of the microfluidics, how the instrument and the CDs work. In terms of Gyros Protein Technologies as a company, here's a little bit about our history. So Gyros AB was originally formed back in 2000 as a spin-off from Amgen Pharmacia Biotech. Globally, we have over 160 employees. We have 2 centers of excellence. So Tucson, Arizona, which is very much where our peptide expertise is, and Uppsala, Sweden, which is where our immunoassay experience is. And both sites are ISO 9001 certified. We have expertise in both microfluid immunoassay development, as well as the manufacture and of instruments to do peptide synthesis. Our customer base is throughout drug development, pharma, biopharma, CROs, and all the way to academics and government institutions. We have a global network of service and support, and we have a distributor network throughout the world so that we can support instruments throughout the globe. And we have 3 sales offices, one in Sweden and also the US and in Japan. Now I will give you an introduction to GyroLab technology and the nanoliter scale immunoassays that can be used for time critical workflows. The core of the GyroLab technology involves the CD or compact disc. This is the equivalent of a microtiter plate where the immunoassay reaction happens. And we have various ranges of CD involving different volumes, but amongst the CD we have CD microstructures where the immunoassay happens, and this involves precise volume definitions. So samples and reagents are transferred to the microstructure as part of the instrument. At the bottom of the microstructure, we have a 50 nanoliter affinity flow-through column where the immunoassay reaction happens. This allows us to have assays of different assay formats. It's very much an open platform, so things like competitive sandwich and other types of assays can be performed. And then detection involves laser-induced fluorescence, and this produces a fluorescent profile, a column profile, and this can be seen in the viewer, and it gives an indication of assay binding. And because detection involves laser-induced fluorescence, this gives us good assay sensitivity, but also a broad dynamic range. So the characteristics of the Gyrolab immunoassay platform is fully automated and gives an integrated workflow. Detection involves laser-induced fluorescence, which, as mentioned, gives us a broad dynamic range but good sensitivity. The volumes are nanoliter scale, which will save you both reagents and sample, and also gives you short turnaround times, which is really good for things like assay development, as a full assay will take 1 hour to perform. So the advantage of using the Gyrolab platform for immunoassays, it involves nanoliter scale consumption of both reagents and samples. It gives a broad dynamic range and reduces number of dilution steps in the assay. It involves automation and enables increased productivity and reproducibility, and also reduced matrix effects facilitate assay transfer in all stages of drug development. As I mentioned, an assay takes approximately an hour to perform. The platform is an open platform which gives very much a flexible approach. It can support many different assay designs, sandwich assay, PK assays, bridging assays, indirect assay formats. We can have a 1-step, 2-step, 3-step, 4-step and 5-step, for example. And all these methods can be downloaded from our user zone. So our end users have a lot of flexibility how they use the platform. So there are many different components of the Gyrolab immunoassay platform. So we start with the systems, the instruments. So we currently have 2 instruments. We have a Gyrolab Expand, which is a 5 CD instrument, and the Gyrolab Explore, which is a single CD instrument. Both are very equivalent to each other, so assays developed on one can be transferred to the other. They both have the same run times in terms of per assay at approximately 1 hour, and they are designed to operate in regulated environments because they're 21 CFR Part 11 compliant. So we have the software also, which is allows us to manage, control and evaluate data, and also plan and do curve fitting, for example. We also have particular modules for things like LIMS integration to Watson, for example, but also modules for things supporting anti-drug antibody assays, and also things like affinity. And all our software is CFR Part 11 compliant. We have a range of different consumables, different compact discs, and the number is relating to the volume. So a 200 is a 200 nanoliter CD, 1000 is a 1000 nanoliter CD, and a 4000 is a 4000 nanoliter CD. We also have high capacity CDs. So these have different high capacity columns, and these can be very useful for different assays, particularly when using polyclonal antis as capture. We also have a particular mixing CD, which I'm very excited about. This is mainly being used for anti-drug antibody assays, where you want to acidify the sample, but it's really good for pre-incubating your samples within the CD, and it can very much increase the contact time. We also supply a wide variety of different reagents. So we have buffers, Rexit buffers, that have been specifically designed for the Gyrolab system and really help the microfluidics and really help with assay development. And I would always suggest that you test the buffers when developing assays on our system. We also very much use moving towards doing Gyrolab kits. So these are ready to use solution. We have a wide variety of different kits, particularly in the cell and gene therapy and bioprocess area. But we also have kits for things like generic PK, TK, and we're very much moving into the biomarker field with ready to use kits for cytokines, for example. We also can do custom kits for you if you want to your assay converted to a kit, we're happy to work with you on this. And we also have a functionality kit, which is very much used to test the performance of the assay and is used for the routine maintenance, for example. And we also have a specific wash buffer that's been developed to minimize carryover, and also it can very much help with your assays. Over the last year or so, we've implemented a new Gyrolab assay service. This has very much come about due to demand and requests from our customers. So we can offer custom biomolecule labeling, so we can take your reagents and both Alexa labeled and biotinylate and provide you these reagents with a certificate of analysis, and we can fully characterize them based on your requirements. If needed, we can also do custom assay development, so we can take the labeled reagents and actually come up with an assay format with you, and we can transfer this assay back to you. And in some cases, we can also move forward to a custom kit development where we can fully develop a kit for you and supply you with a finished kit that you can use in your lab. And in summary, please put Gyrolab technology to work. It very much can accelerate assay development and sample analysis for both preclinical and clinical research and development. It can very much facilitate upstream and downstream bioprocessing analytics. It can meet critical data requirements for regulated bioanalysis, and this is very important to us. Most of our customers we work with are very much in the regulated area. It can strengthen quality control in manufacturing, and whatever your immunoassay requirements are, Gyrolab technology can help you get there faster. Thank you everybody for listening, and now we're going to move on to Issa's great presentation.
Hi and welcome. My name is Issa Jamurandi. I'm a principal scientist here at Drug Development Solution, a CRO contract research organization based in Cambridge, UK. Cell and gene therapy has been one of the hot topics over recent years, and we feel that this is a perfect time to be discussing some of the challenges that we encountered while developing immunogenicity and quantification assays for some of the transgene enzyme products, especially using the automated platform, the Gyrolab. So I'll go through a quick overview of some of the points that we'll cover throughout this presentation. First, we'll go over a quick background about AAV in general, as that's the main point of discussion today. And then we'll go down to talk about some of the quantification challenges that we encountered. And in addition to that, we'll share some of the platform comparison data that we generated so far. And that helped us in terms of determining what was the best platform for some of the examples that I will share. And then we'll move on to talking about immunogenicity assessment, also using the Gyrolab platform of some of the transgene products that we've looked at. At the end, we'll provide some of the conclusion and some future recommendation in terms of assessing PK and immunogenicity of transgene products. So to start, let's look at the overview of AAV gene therapy in bioanalysis. For the purpose of this talk, as I mentioned, we'll focus on adeno-associated viruses. Adeno-associated viruses are replication-deficient, non-enveloped viruses. They're very promising currently because they're mainly very low immunogenic and also have demonstrated long-term gene expression. AAV are able to transduce a wide variety of tissues. There is over 11 well-known human viral serotypes, and AAV are not known to cause any human diseases. The mode of action of AAV, they enter cell via endocytosis in a cell in a receptor-mediated manner. After endocytosis, the AAV vector escapes from it, the endosomes, and enter into the nucleus, followed by the release of their single-stranded DNA genome, which in the absence of a helper virus, they will remain in the nucleus in an episomal state. And in terms of bioanalysis, the key part of what we are really interested in are mainly before the AAV gets into the host cell, we're more interested to know whether there is anything that prevents it from getting into the cells. And by that, I mean, we're looking at the immunogenicity, pre-existing antibodies against AAV. In some of the assays that we've developed, we've demonstrated that there's, in some cases, over 70% of pre-existing antibodies present for some types of AAV. So it's important that those are pre-screened to avoid the presence of those candidates in a trial, or at least introduce means such as the removal of those pre-existing antibodies, selectively remove those pre-existing antibody before introducing the patients into the trial. Once the AAV are into the host cells, the other key component to consider is we need to monitor the DNA or the genomic material that comes from the AAV, and that is generally done by qPCR. But the purpose of this talk will be mainly on transgene product, where we'll be focusing on the measurement of the transgene product. So in terms of that, I refer to quantification of the transgene product, and also looking at the immunogenicity profile of those transgene products. So if we focus on the transgene quantification first, when we hear the terms quantification, the first thing that comes to mind is PK. However, when you think about transgene product, it's very clear that they don't actually fit within current PK guidelines. And the main reason for that is the fact that they lack a true reference standard. And the lack of such a reference standard means that the only quantification that can be done on this transgene product is relative. What has to be done to be able to quantify this product is the use of an alternative available therapy that mimic expected transgene as a calibrator. And because of that, we feel that the measurement or the quantification of transgene product fits better as a biomarker in the biomarker assessment world more than a PK assay. So as a result, we feel that the context of use is more important for transgene assessment. Therefore, where we be more interested in looking, for example, for the change in baseline to physiological relevant levels. And one of the most important things to consider when developing this transgene product is that most of the transgene product will resemble the endogenous protein. As a result, it's very hard to discriminate between the endogenous molecule and the transgene product, which is why if you know in your transgene product there will be some sequence dissimilarity between your transgene product and your protein, we would always advise to use the LCMS method, which will be able to discriminate between those two. However, we all know that the LCMS method might lack some of the sensitivity that a ligand binding assay might be able to provide. Every case, parallelism should be important, as especially in a ligand binding assay, the calibrator used in this case will not be a true a reflection of your transgene product. Parallelism becomes key. So therefore, the assay is only fit for purpose and only becomes really relevant once the sample has been assessed and parallelism has been demonstrated in sample containing transgene product. So in terms of transgene quantification, there are some of the key components that needs to be considered. Especially the sensitivity is very key, as already mentioned in my previous slide. Sensitivity of some of these transgene products can be a very low nanogram or picogram range. However, one of the good things about this transgene is that you expect them to be consistently expressed at a certain level. So therefore, the dynamic range of the assay required is not very large. In most traditional PK assay, a stepwise format is mainly preferred, mainly because you want to avoid hook effect. However, in cases of transgene product where the assay range is not large, homogeneous assay format could be accepted, and homogeneous format are well known to be able to provide the best selectivity and the most amount of sensitivity. And in some of the cases that I will describe later, it's also been demonstrated to provide the best parallelism compared to a stepwise format. So when working with transgene enzymes, it is one of the most important things that we found is that the buffer plays a crucial part in developing these assays, as buffers can have huge impact on conformational change of some of these enzymes. So on my next slide, I can show you some of the buffer impact on the enzyme. So if you can see on this graph, we looked at the assessment, for example, of one of the transgene enzyme in some of the Rexit buffer, and also included some of the common buffer that we use, like 1% casein and 0.2 casein and low cross buffer. And as you can see on this graph, Rexit buffer, for example, which are well known to work really well for things like antibodies, were not perfect match for enzymes. And we postulated that that was probably something to do with the salt content of Rexit buffer. While those buffers are very good for antibodies, they might not actually be very good when it comes to enzymes. However, when we use simple buffer like casein, you can see that casein improved the sensitivity of the assay. However, the only problem with casein, it has very poor solubility. When we carried on using casein, we noticed that the precision in the assay was relatively poor. So while casein might be very good in terms of improving the assay sensitivity, we also incurred problems in terms of precision in the method. So it's important to consider when developing these assays, things like temperature, pH, and salt. When it comes to enzyme, these are things that are also important for all the other analytes, but becomes even more important when it comes to enzymes, as all these three things mentioned above here can play a key factor in terms of conformation or change of the enzyme. And when you change conformation, you can lead to epitope changes or even masking, which can lead to various different recovery, poor, even higher recovery in some instances. So one thing that we've learned throughout this whole project is that some of the best way to create the surrogate matrix is to treat your matrix at 56 degrees, which means that you actually denature all the endogenous enzymes and end up with a matrix that is deficient with that particular enzyme, and that matrix more mirrors the patient matrix than actually using the surrogate matrix. And in this case, we also demonstrated that treated matrix improved parallelism compared to the use of any other surrogate matrix. So the next slide here was more of sharing some of the platform comparison that we also did to show that the Gyro lab in this case was slightly better than the actual use of the MSD. We carried out some of the simple experiment, such as the sensitivity assessment, and we actually demonstrated that the Gyro lab was slightly better in terms of sensitivity. And we also demonstrated that the precision on the Gyro lab in this particular case was slightly better. And the most important thing, as mentioned earlier, was parallelism. And when we did this experiment on both platforms, we also showed that on the Gyro lab, we had much better parallelism data compared to what we had on the MSD. And that was very good to see. And we then decided to actually carry on working on the Gyro lab in this particular instance. So some of the key take-home message from this quantification methods that we developed are mainly that quantification of transgene products do not actually currently fit with PK guidelines that are already available. So it's more aligned with biomarker assessment. We feel that the context of use is more important in this case, and the method will be validated to be fit for purpose for predefined context of use. When developing these enzyme quantification assays, it's important to take into consideration temperature, pH, and salt, because they can have a detrimental effect on your enzyme and therefore might mean that your quantification that you're carrying out, whether of your samples, might not necessarily be accurate. So it's important to take into consideration those factors. In terms of developing methods, whether you go down the route of ligand binding assay or whether you go down the use of LCMS method, when you develop these methods using ligand binding assay, it's important to remember that the ligand binding assay might not necessarily be able to discriminate between your endogenous molecule and your transgene product. So as a result, parallelism is very crucial to demonstrate that the ligand binding assay method is fit for purpose. And that is the end of my quantification part. So the next part is the assessment of ADA method for transgene product. The same as the PK assay, there's no guidance currently on immunogenicity assessment of transgene product. The current FDA guideline clearly stipulates that it doesn't relate to the cell and gene therapy product. So while developing these methods, we had to look elsewhere to find what might be more appropriate to assess these immunogenicity against this transgene product. So if you look on one of the FDA document for human gene therapy for rare diseases, they clearly stipulate that immunogenicity for AAV and transgene product should be assessed. And also, when you look at the FDA guidance for human gene therapy for hemophiliac, they also go on to suggest the long-term monitoring for immunogenicity should be done for at least over 2 years. And they do suggest to monitor that immunogenicity for the vector and for the transgene product periodically, and also go on to suggest to do some cellular immunity, which is why internally we've adopted the same approach of assessing transgene products the same way as you would assess for a normal protein. First, initially by screening any sample, and if any sample is positive, you take it down the route of a confirmatory assessment. If it's negative, obviously report it as negative. And if it confirms positive, you also need to determine the magnitude of antibody presence in the sample. And that's where we do experiments, a titer experiment, which would then be followed on by NAbs assay to determine whether there is any neutralizing antibody present in those samples. But one important consideration to consider here, as you can see with another arrow that comes from the screening straight into the NAbs, is that in some cases, because of the conformational change of the enzyme that may occur while doing conjugation or with some of the buffers, it's important that you assess whether any of those conformation change didn't affect the enzyme so that the catalytic unit is not affected, that you're not able to detect that in the ADA assay. And if there is any evidence that that's the case, then it's important that any sample that's analyzed in the screening assay, in the ADA assay, is also analyzed in the NAbs assay, just to avoid missing any NAbs positive sample. So some of the initial consideration that we had when developing this method was, especially on the Gyro lab, we've got the flexibility of developing a method on a homogeneous or stepwise format. We had the choice of doing any of these methods, but when one of the very good key point that we knew before starting developing this method is that the transgene endogenous circulating concentration is present at very low level. So any of these methods, whether homogeneous or stepwise method, could have worked. One of the other consideration, as discussed in a previous slide, is when you conjugate these enzymes, you want to keep them in the right medium, just to make sure that you don't end up masking any of the epitope or ending up with a higher conformation change in conjugated reagent that you might not be able to detect any neutralizing antibody in the ADA format. In terms of Gyro lab platform choice, at this rate, we were already sure that we were going to use the Gyro Lab for determining ADAs in this assay. We got the Gyro Lab Explore, which uses one CD, and we got the Gyro Lab XP or Expand, which uses more than one CD. And given the example that I described earlier with buffer and incubation, having multiple CD can have an impact, and that's what I will discuss on my next slide. So the initial work that were done were carried out on the Gyro Lab Explore, where one CD was used throughout, and the condition was selected and the method was sensitive enough. So when the conditions were selected, the capture was dissolved in RexipEF, the detection in RexipEF as well, and the sample diluted 5-fold in RexipADA. However, when the method was then transferred on the Gyro Lab XP, where we use more than one CD, and in terms of an ADA, because you need to do cut points analysis, you need to run at least at least 3 CD at a time. We observed a signal loss over multiple CD, and as you can see here, CD1 had higher signal, and as we went along all the way to CD4, you had very huge deterioration in signal, which would have resulted in CV issues. So we then started an investigation on what caused these issues. So first, we approached the Gyro Lab. The team were very friendly, and they helped us in terms of initially determining which buffer was appropriate. And the first point of call was looking at the buffer that had an isolectic point that was closer to the enzyme of interest that we had. And they suggested the use of RexipHX. However, one thing to bear in mind is that the enzymes, as demonstrated in the PK or discussed in the PK, the enzymes are not just tolerant or sensitive to just various pH, they're also sensitive to salt and temperature. One other important consideration is that when we were having this discussion with Gyro Lab, is that these buffers are proprietary to them. So it's very hard for us to troubleshoot it if we actually don't know the constituent of these buffers. So we had to go with their recommendation that they were giving to us. So we tried the RexipHX as it was given to us, and we saw exactly the same thing, signal deterioration over multiple CD. And the other thing that we noticed with this is we incubated the samples at room temperature for 6 hours, and we also noticed a loss of signal with samples that were pre-incubated in the dilution buffer for 6 hours and the conjugated reagent for 6 hours. So the effect was clear that it was due to the buffers that we were using at the time. Just to test all the other buffers, we also tested RexipADA to use it as diluent for the samples and also diluent for the conjugated reagent. And we also observed exactly the same thing in terms of loss of signal over time. The other thing that was very apparent throughout these experiments is that the loss of signal was also associated with higher precision failure. So because of that, we thought, given that the buffers are the root cause of this, we went back to a simplest buffer that comes from the Gyro Lab, which is the wash solution one. And by the use of wash solution one, we actually saw an increase in baseline, which led to poor sensitivity, but it was clear that the increase in signal also improved the CVs. But this was very good data because it was clearly demonstrating that the issue was with whatever was in the Rexip buffers that was causing issues with the enzyme. And that helped us in terms of optimizing other buffers that we could have used. So we went back again to the basics by using casein, which has also been demonstrated in the PK to work. And casein was very good in this case, that it improved the assay sensitivity, but also reduced the baseline to the levels that we wanted. However, the only issue, as discussed in the PK as well, the precision was an issue for casein. So again, we approached the Gyro Lab team, and they were very good in terms of advising us on the best course of action, which was first to try and increase the centrifugation to maybe 14,000 G for samples. And also, after applying that, we still saw some of the CV issues. And as an additional thing, we had to go back to the method again and ask JaraLab to design a method for us, which would include a mixing step. And the inclusion of such a mixing step on the JaraLab improved the precision issue from more than 6 CV failures per CD to less than 3 CV failure per CD. So the use of these steps improved the assay precision, but it didn't really abolish completely, but at least we were in a reasonable number of CV failures that we observed on per CD. So to conclude, while developing these assays, it's very important to remember that they are very sensitive to temperature, pH, and also to salt. So it's important to take those into consideration. And we've already spoken to JaraLab, and they're already working on developing some of the buffers that might be suitable for these particular enzyme. So the other important consideration is that when conjugating these enzymes, it's important to keep them in a happy medium to avoid masking or changing the conformation of the enzyme to the point where you might end up not being able to detect any neutralizing antibody in the ADA assay. Where that happens, it's important to make sure that the conjugated reagent are tested in the potency assay or in the neutralizing antibody assay to make sure that you're still able to have the right conformation, and also that you're able to detect all the antibodies in your ADA assay. And finally, which is a learning point for nearly every assay, before transferring from the JaraLab Explore to the JaraLab XP or even Expand, it's important to test extended sample stability and reagent stability to make sure that the assay can run reproducibly over multiple CDs. And as a final recommendation from us to the JaraLab, it would be nicer if all these issues could have probably been resolved if the JaraLab was able to spin 5 CDs at the same time, and actually also add the reagent at the same time. And that would have avoided the issue of signal deterioration, because currently the CDs are being run each at a time. That would also lead to shorter run time, which will be a success not only to the customer, but also to the JaraLab. So if a human can do it, surely an automated system can do it. So thank you for your attention. And I'd like to say thank you to these people who helped develop the method. Tom, who worked on the PK side of the method, Richard Hughes, our Associate Director, and Debbie for all the input in terms of the method troubleshooting, and the entire DDS team, method development team for the input in developing these methods, our DDS marketing team, and the JaraLab technical team for some of the input during the method development. So thank you.
Well, thank you to Issa and John for some great presentations. And as Issa mentions, we're now going to start the Q&A portion. If you'd like to both join me back on camera.
Hi.
There we are. Okay, fantastic. So the first question I've got here is for John. So regarding the custom assay service, how long does it take to have an assay designed?
I think that's a very good question. I'd say, though, it depends on the scope of the project and also the nature of the assay developed. Obviously, once a legal agreement's in place, I mean, assay development can be very quick. I mean, I've seen anything from 2 to 3 days to a couple of weeks, but it really depends on the scope. We very much want to work with customers to get assays developed as quickly as possible.
Fantastic, thank you. And Issa, the question is for you. In the steps described for the ADA, there was no mention of acid dissociation. So was this used? And if not, how was the required drug tolerance achieved?
Oh, thanks for the question. I think that's a brilliant question. In most of the ADA assays, we tend to add an acid dissociation, mainly to improve drug tolerance. But in this particular case, when we actually tried the use of drug tolerance, we observed that the signal baseline was actually significantly going higher. And we actually, when we looked at the data back, we found that potentially the reason for this happening was that the acid and base were probably doing something to the enzyme, causing aggregation and causing the baseline to go higher. That's when we had to go back and revisit. And actually, we noticed that the heat was denaturing the enzyme as described in the PK assay, and therefore we just use the heat as a means to make the assay more drug tolerant or transgene tolerant. And that was a winner in this particular case. And if we wouldn't have had to use the heat, I don't think we would have achieved the levels of drug tolerance that we achieved, which was up to 1 microgram per mil, and that was way above the required level by the sponsor.
Fantastic, thank you. Now, I've got an audience question, which I think we're staying with you for, Issa. It's a bit of a long one, so bear with me. What is the preferred approach to doing transgene product PK assay, considering it is more appropriate to develop this with relative quantification under biomarker type of assays? So use surrogate heat-treated matrix to eliminate endogenous counterpart, or to use regular matrix and use the assay as relative concentration changes to over time from baseline. Of course, the later option would only be possible if the endogenous levels do not change from baseline post-drug administration.
So, I mean, the use of that surrogate heated matrix was actually a winner again for us. So the option was either to buy a surrogate matrix or to use this surrogate matrix, which was heated. And actually the use of surrogate matrix here, when we heat, the advantage of that is you're removing all the endogenous matrix and keeping the matrix that is potentially similar to a matrix that is deficient in the transgene product that we'll be looking for. Some of the advantage that comes with that is you improve the sensitivity of your assay, and also the other matrix interference that you will actually see in your study sample are still there, so you can resolve them at the same time. One of the surprises you don't really want to get is when you get into sample analysis and realize that you have poor parallelism, because that's one of the tricky part here, is that you always have to remember that the transgene product is not what we're using as calibrator in this case. So when it gets to sample analysis, you want to make sure that all the matrix interference that you've seen in the development are hopefully similar to the ones that you're going to see in your sample analysis. So some of the advantages illustrated is that you improve sensitivity by using heat-treated matrix, and that's why we actually went ahead with the heat treating of matrix.
Fantastic, thank you. And Issa, I've got another one for you here. So you mentioned that the method for transgene quantification assessment can only be classified as fit for purpose after sample analysis. Can you elaborate a bit on that, please?
So yeah, so trying to elaborate on that again, that comes back to the use of a reference material. So if you think about it, most of the PK assay, we have to use a reference material which is well characterized and have the right paperwork. But in this case, obviously, we're using a product that mimics expected transgene product. So in that case, the quantification that we're doing and all the validation that we're doing are not exactly of what we expect for the transgene product. So in this term, we are always going to claim that the assay is 100% fit for purpose once we get the samples that contain the actual transgene, quantify them, and actually also carry out the parallelism to make sure that the dilution of these samples mirrors that of the surrogate product that we've used to create our curve for our validation and to show that the method is fit for purpose.
Absolutely, thank you. And I've got another one for you here. When developing the transgene quantification assay, you learned that you received slightly higher sensitivity, better intra-assay precision, and parallelism with the GyroLab platform compared to the MSD platform. Have you experienced these differences in result between the technologies for other types of assays? And how long did it take to develop the respective assays?
So in terms of development of these assays, actually, most of the PK assays that we have internally we've adopted the principle whereby when we're still in the development phase, we'll always try and develop these assays on multiple platforms, just so that we can get a better choice in terms of what platform fits the assay better. And in this particular case, the Gyro Lab was better than the MSD, but it's not always the case. But certainly one of the biggest advantage with the Gyro Lab is you can develop your assay very quickly compared to the use of the MSD. But we have seen in some cases where the MSD can give you the required sensitivity. But again, in this case, even though the MSD could have probably provided better sensitivity, the method was not parallel. So the Gyro Lab provided more data in terms of parallelism, which was actually key in terms of what we were looking for here.
Fantastic, thank you. And I think this is another one for you, Issa. Could you discuss further why the homogeneous assay format was beneficial for PK analysis when compared to the stepwise assay format?
So in terms of the homogeneous assay format, in this case, actually, the homogeneous format, in any form of a PK assay, when we look at most of the PK assay, we'll always prefer to use a stepwise format. And most people will know that the stepwise format is key in terms of avoiding things like hook effect. And in terms of a PK, you've got a very wide dynamic range in some cases, and you don't really want to be underestimating the concentration of your drug in the sample. But in this particular case, we knew that the dynamic range of this assay is very narrow, and homogeneous assay are very specific. And in most cases, actually, we demonstrate that it can be very sensitive as well. And in this particular case as well, it actually proven to be the most sensitive method, and also it was more specific. So the parallelism on the homogeneous format was much better, and that also comes back to that specificity as well. And as a result, we chose the homogeneous format. We also investigated the stepwise format, but actually in this case, the stepwise format, but in this case, the homogeneous was much better.
Fantastic, thank you. And John, I've got one here for you. Are there any guidelines available to know which type of CD to use depending on the assay and the strain?
I mean, it really depends on what the assay sensitivity requirements are. We'd probably suggest that most of the time that assay development could be started on either the 200 or the 1000 nanoliter CD. This ensures that you get an assay and you can screen reagents and get test the assay range. If you need a very sensitive assay, we would recommend that you use the 4000 nanoliter CD. And if you really want to measure very high concentrations, you'd probably look even at a 20 nanoliter CD. The advantage of a CD such as the 200 nanoliter CD, it has additional structures, 112 structures. So when it comes to reagent screening, it allows you to screen more reagents when you do checkables, etc. So it really depends on the assay. But as a rule of thumb, start with probably around the 1000 nanoliter CD as a good choice for initial development.
Fantastic, thank you. And I think this one is for you, Issa. For the ADA assay, what was conjugated since you do not have a hold of the enzyme product itself? Was it a surrogate enzyme?
Yes, so in this case, we again, it's the same problem. The same problem for a PK assay is the same problem for an ADA. So you have to sort of find something that will mimic your transgene product, and then hope that the transgene product will be the same as your surrogate product. Obviously, we're still early stages at this stage, and the best advice will be to get the exact product that you want, the transgene product, and find it expressed maybe in an animal or somewhere else, so that you've got the exact transgene product, and that would be the perfect case scenario to be in, and that would be the same perfect case scenario for a PK. But I think at this stage, we're trying to advance the research, and it's appropriate at this stage, I think, to us to use a surrogate. But I think, as I said, the perfect one would be the use of the transgene product.
Absolutely, thank you. And another one for you here, Issa. What % casing worked for the ADA?
So percent casing, we actually use 1% casing. One thing that is worth bearing in mind is that casein at 1% or even 3% is still not very soluble. So the solubility of casein is very poor. So if you leave it overnight in the fridge, next morning you see some precipitate. So 1% would work, but you still need to implement some other strategies such as centrifugation, higher centrifugation, and making sure maybe that what we asked the Gyro Lab to help us with include some mixing step to be able to actually achieve desired precision in the assay. But 1% worked, and it gave us desired baseline and also improved the precision issues that we were having with the additional measures that we had to implement.
Fantastic. Thank you. Well, thank you, Issa and John, for answering those questions. I'm afraid that's all we've got time for today, but any questions we didn't get to, we will reply by email. The webinar will be available on demand from tomorrow, so look out for an email from us with the link. All that's left is to thank our presenters once more for a great presentation and discussion, and thank you to the audience for listening. We hope you'll join us again soon.
Thank you.
Thank you, everyone.